So again, thanks so much, Uma, Ansil, and Farah for having me. I just realized that I didn't actually thank anybody in my first talk, so making up for that now. But yeah, so today I wanted to kind of, again, so last time, of course, I just did the chalk talk, and so I wasn't able to present any of the data and everything else, but this is a slightly different take. I wanted to present this data. It's very preliminary, so I was I'm interested in getting feedback on what your thoughts are and everything else. So you already learned about these species distribution models. Farah was talking to you about genomics and how we can use genomics in looking at vectors and everything else. We've also been, and Kavita today in the morning was talking to you about climate change and how it's going to be affecting vector distributions and everything. So this is the basic kind of framework within which I'm going to be talking today. So this spatial mapping of these parasites and vectors is becoming an increasingly powerful tool that we are using. And these kind of maps can be, these are these kind of deterministic models. I mean, we can use them to actually find out, what the current fundamental niche of these parasites are. And then once you have a set of environmental variables, you can actually predict where these species are going to be going. So these species distribution models, for example, are a very, very powerful way of going about doing these things, right? You can also include other aspects. For example, you can include things like human movement, right? You can include how disease control is going to be affecting. So these are kind of more the geostatistical models. I'm not going to be talking about those today. I'm mainly going to be talking about this kind of environmental modeling and these so-called species distribution models, and not only kind of like how they're being used, but mainly what their weaknesses are. Okay, so when we're talking about these species distribution models, I would say that one of their strengths is the fact that you can predict what the future is going to look like, and therefore it'll help us to kind of decide what the future control is going to be or where the disease is going to be in the future. Okay, so how do these models work? Again, you have already gone through this, but I just want to go through this just to make sure that we're all on the same page. So we mainly use these rasters, like what was mentioned yesterday. So these are these GIS files, right? And so each of these grids or cells or pixels or whatever it is will have a certain value associated with them, and that could be elevation, it could be forest cover, it could be anything, right? So you have these environmental factors. Then you have so-called these presence or absence grids, right? And so again, we are only talking about species distribution models. We're not talking about those occupancy models that were being mentioned earlier. So we are assuming for the time being that the places where you have a 1, you truly have a 1. The places where you have a 0, we call them pseudo absences, in the sense that we don't have data that they are absences, but we know that at least we haven't, all the time that we've gone and seen, we haven't found a tick there. So basically, if you have a 1, you know that there is a tick, but just finding a 0 could be due to 2 different factors. One is that the tick is there and it wasn't detected, or it's not there at all, right? So that's fine. And you use both of these you use like these statistical models to predict the habitat suitability for that particular vector or species of interest, right? So usually you're going to be using these kind of machine learning approaches, random forests or some approach like that, but we are not going to get into all of those details. For the purpose of this study, we do use the random forest, right? Okay, so then if you have some kind of an environmental gradient, right, that is determined by all those environmental variables, you can predict this kind of distribution of this particular tick, right? So then you can say that, because of climate change or whatever it is, this envelope, this kind of climatic suitability envelope is going to be moving, right? And so you would assume that the tick would move with that climate suitability envelope, right? So it's a very simple kind of a prediction. The problem, right, is that increasingly we are getting evidence that it is not just that these ticks are not only kind of tracking climate events, but they're, did you have a question? No. They're not just tracking climate events, but they are actually being moved around by humans, right? And they could adapt to new situations, right? Because these are like living organisms. So the traditional species distribution model kind of assumes that the tick itself doesn't adapt at all, right? So for example, in this particular case, what you would say is that, okay, fine, this is a tick. If it was just moved, like just physically moved by a human or by a deer or something of that sort to a new environment, you would predict that that tick would just kind of disappear, right? It would not survive. However, we are increasingly finding that, actually speaking, the ticks could adapt. They could become a slightly different tick, and therefore they could actually survive in a new environment, right? So the question that we set out to address in this study was, can we actually incorporate dispersal and evolution into these species distribution models so as to predict the range expansion of these ticks better, right? We've used this model for several species now. So again, as I told you, it's a very preliminary model. We are still waiting for some data and everything else. We've used it for mosquitoes. We've used it for an invasive plant. It seems to be doing pretty well. But again, the limitation I will come to right at the end. Okay. So, so where did all of this start, right? It started because when I was in the US, so I was at the University of Georgia, I was very interested in this tick, which is Amblyomma americanum, right? And we got interested in this tick because it was, it's causing this, what is called a red meat allergy, right? And so when the tick bites, you get this bizarre allergy to red meat, right? And it is being driven by this alpha gal, which is basically a carbohydrate, right? And so it's a very fascinating allergy because there are very few allergies to carbohydrates. Most allergies are being caused by proteins, right? So it's a fascinating kind of a situation. And it has been shown by multiple people, including us, that basically this alpha gal is the causative agent for this thing, and the Amblyomma americanum has this alpha gal, right? So if the Amblyomma americanum bites you, you get this alpha gal into your system, it primes the system to react to alpha gal in meat or something like that, and you get this strong kind of anaphylactic reaction. Okay, so we were very curious. So there was a paper that came out in 2014. So this is the this is the data that we've been using because 2014 is like the best kind of known distribution of this tick, right? But the paper was also showing what was established in 1930, right? So the dark green areas are, so this is county level data. I think in India, what is it? It's your district, right? Yeah. So this is almost like the districts in India, right? So this is county level data, and you can see that this is only established in 1930, established in 2015, or reported in 2015. The only difference between established and reported is that established means there are multiple reports of that tick from that same county. Reported means that there are only one or two. It may be, accidental report or something of that sort. So you shouldn't take that data very seriously, right? Okay. Now, you can see that, so this is Virginia, right? And you can see that as of 1930, the northernmost distribution of this tick was in Virginia. But by 2015, you can see that it has gone into New York and even into parts of Connecticut, right? So basically, it has moved quite a bit up north, right? Okay. So so the great thing about these species distribution models and everything else is that usually what happens is that we take the data for now, and then we make these predictions for like 100 years in the future, right? So the only way that I can be proved wrong is for somebody to wait for 100 years and say, well, actually your model was completely wrong, right? The question is, how good are these models really, right? So since we had like the 1930 data and we had the 2015 data, we did this kind of a thought experiment where we fit the species distribution model for the 1930 data with the 1930 climate. Then we used that model to predict where the tick would be in 2015, right? And this is exactly what everyone is doing. Mainly they're using the current distribution and the current temperature or the current climate to predict where the tick will be in the future. But obviously we don't know whether that's true or not. But now we actually know how good these models are, right? Okay. What did we find? We found that the model predicts this would be the upper northern limit of this tick, right? So the 1930 predicted limit and the 2015 predicted limit, right? And actually speaking, if this tick was moving only due to climate change, this is probably going to be correct, right? So there was a paper that came out in Nature, this Parmesan and Yo, and what they did was that they basically looked at, how climate affects the movement of species across a large number of species, and what they found was it moves at about 6 kilometers a decade. So basically, you're going to be moving northward at about the rate of 6 kilometers a decade. In our case, we have about 85 years, so about 50 kilometers, right? So the model is actually predicting the movement due to climate change reasonably well, at least in line with what we expect for a large number of other species, right? But where is the tick really? Well, the tick is all the way up to Connecticut, right? So this is the predicted, this is where it is, New York, parts of New York, and at least it's been reported in parts of Maine. So you can see that we are not talking about a small error, right? We're talking about like an error of like 80 or 90% under prediction in terms of this model. Okay, so what did we start? So the first thing that we thought about was that, well, actually these ticks, they are being moved large distances by these deer, right? So the deer is the final host of these adult ticks. These deer can actually move long distances, right? Humans can move ticks around. So if you go to Florida or something like that, and you pick up a tick, and then you can drive all the way up to Maine or something, and then the tick can fall off there, right? So humans, deer, so the dispersal of these ticks could be really large. So if, for example, you do pick up a tick that is adapted for a slightly warmer climate like Virginia, and then you drive all the way up to, say, Connecticut or something of that sort, will the tick survive? We don't know. It depends upon the genotype or the genome of the tick, right? It is possible that just by random chance, right, you have a tick that is adapted for that climate, and if you have that kind of a tick, you could have a situation where that population establishes in that particular area, right? So what we said was that let us make a new kind of a model. We call this evolutionary moving envelopes to predict range expansion, or the emerge model, right? And it is mainly focusing on how species are going to be, or vectors are going to be emerging, or diseases probably going to be emerging in the future. And this model has three major components. We have a species distribution model, which is going to be modeling that core niche of the tick, and that's just a basic species distribution model with some small modifications which we'll come to. We also model dispersal of the tick using landscape resistance. And then I think what is the most, in my mind, the most unique aspect of this model is that we have this evolutionary genetics or evolutionary genomic framework, right, which allows these ticks to adapt or to evolve to new climatic conditions. The greatest advantage, in my mind, of this evolutionary genetics framework is that this model now makes spatially explicit genetic predictions, right? So one major issue that I've always had with species distribution models is that how do we validate these models? We validate these models with what is called this 7-fold or 10-fold cross-validation, right? So what we do is that we build the model with 70% of the data, and then we hold back 30% of the data, and then we ask, once you build the model, how well does the model predict the rest of the 30% of the data, right? This is how we usually do it. The issue is that the data that you're using to validate the model and the data that you're using to build the model are exactly the same, right? This is fine when you're talking about current, right? If you want to predict for the future, right? So I think, was Farah talking? Somebody was talking about this thing of extrapolating, right, beyond the model, right? So when you're predicting for the future, you are extrapolating to beyond what you built the model for, right? So it'll be nice to have a way of validating the model which is completely independent of the kind of data that you're collecting to build the model, right? And in this case, since we have explicit population genetic spatially explicit genetic predictions, we can actually go out, do genomic analysis at large spatial scales and ask whether this model is predicting it correctly, is not predicting it correctly, and then tune the model so that we get better prediction. Yeah, okay. So, so I'm going to skip a lot of the model details. I'll only give a very, very brief kind of thing about what the model details are all about. But basically, we just start off with this kind of species distribution model, which is modeling the core niche of that species. This is in the case of the Amblyomma americanum that we have, the 1930 niche, right? Where was the species present in 1930 with the 1930 climate, right? So this is what we used to start this model running. The one slight change that we made was that, so you have all of these landscape layers, right? So once we built the model, so you can see that there is, so this is what is called the percentage of your mean square error, change in mean square error, and that's basically like, if I remove this variable, how much error increase is there in the model? And it basically is a measure of how important that variable is. So for example, something like bio2, which is your mean variability in your temperature, is not very important. So just because you remove the variable, you don't get a huge increase in your error rates. However, something like vapor pressure in July has a huge increase in your error rate. So it's a relative importance of that particular variable, right? Okay. So you can see that when I do a partial probability plot and I'm looking at, the places where the tick is absent versus the places where the tick is present, you can see that vapor pressure really has a huge effect. So these are all the places where the tick is, I should probably just use this. Yeah, this is all the places where the tick is absent. This is the places where the tick is present. The tick is present in places which have high vapor pressure. And I think somebody was mentioning that ticks are extremely sensitive to humidity. So they can dry out very, very quickly, right? However, you can see that in the case of your bio02, there's really not much difference between places which is the tick is absent and places where the tick is present, right? So you can see that this is pretty much a flat line, and this is a very clear kind of a peak at this particular point. Okay. So then what we did was that we found this kind of the peak temperature, and we said that this is probably the ideal niche for this particular tick, for this particular environmental variable, right? And so then we went back to our environmental rasters, and we basically standardized all these rasters so that the rasters would be 0 at this peak or the ideal kind of thing, and any increase in the value would be positive, and any decrease in the value would be negative, right? Now remember, a positive and negative doesn't mean that that a positive means that it's getting better for you, and a negative means it's getting worse for you. Basically, as you move away from 0, things get worse, right? So, so 0 is the ideal, and then anything away from 0 is worse, right? So you can see that we can actually plot these rasters, and these are the standardized rasters, and you can see that this is the ideal kind of niche. So all these kind of the 0 points are what is kind of ideal. Anything orange is going to be like hotter than the tick prefers, and anything, wetter than the tick prefers, and anything blue is going to be drier than what the tick prefers, right? And the stronger the color, the worse it is. And similarly for our diurnal temperature range, so the more the temperature range, the worse it is, right? So yeah, okay. So the next is that we wanted to look at how these ticks are moving across this landscape, right? And we actually don't have any data on these ticks and how they're moving in the landscape and everything else. So what we ended up doing was that we ended up using like the 1930 data, presence and absence, and the 2015 data, presence and absence, to just see whether or not a particular cell was connected or it's not connected, right? And we used what is called landscape resistance, and I'll tell you a little bit about what landscape resistance is all about, right? So you can imagine that, so what most of these, what the traditional dispersal models used to do was that they were mainly looking at distance, right? So the further you are away from a point, the less likely you are to get. We were looking at isolation by distance, right? I forget who was showing isolation by distance. Was it you, Farah, who was showing isolation by distance? Yeah. Yeah. So isolation by distance is a very common kind of a pattern. But you can also have something which is called isolation by resistance, right? So basically, the more resistant to, or the more resistance the landscape has to dispersal, the less the connectivity that the 2 points will have. So for example, I'm using a deer here because a deer is one of the most important natural hosts for the tick, which has a relatively large kind of a dispersal distance. So you can see that if a deer has to start from this point and end at this point, there are 2 ways, well, there are multiple ways the deer can go, but the 2 ways that I'm showing is, one is just a straight line, and another is all the way around, right? Traditional models would say that the best path is the shortest path, right? But you can see that the cost of going through these different landscape cells, so this is what a raster will look like, right? So each of these are pixels. The pixel is either forest or agriculture or or urban area or whatever it is, right? So what it's saying is that the cost to go through a forest patch is very small, but the cost to go through an urban patch is very large, and the cost to go through agriculture is somewhere in the middle, right? So if you model this, you will see that this straight path has a linear distance of 17, but the cost distance is also 17, right? Because each pixel has only a single cost, right? In the case of the straight path, though the linear distance is much lower, so it's 17 versus 10, the cost distance is very high. It's like 65, right? So therefore, the deer is more likely to take the least cost path, go through forest areas, right, rather than go through all this urban and agricultural land, despite the fact that it is shorter, right? So basically, this least cost path is an important aspect. The problem that we face is that we don't know what are the landscape variables which are going to be permissive to ticks and what are the landscape variables which are not going to be permissive to ticks. So what we decided to do was to build a model that would help us to identify the variables that are important or allow the ticks to permeate, and the landscape variables that prevent the tick from moving, right? So how does this model work? Well, for example, if I have 2 cells in Virginia and all the way up in New York, right? The model starts with a homogeneous kind of a cost path, right? And we basically say, okay, fine, if it's homogeneous, then the least cost path is, of course, the straight path, right? And then in every optimization step, right, the model will evaluate like how these costs are going to be changing till finally you get a cost optimization which will say that, well, actually speaking, the tick is not going to be going straight up like this, but it's actually going to be going along the coast and out this way, right? And then we can actually evaluate what are the variables that are most important, right? So in this case, why does the tick go all the way like this? Because of the Appalachian Mountains right here, right? So there's very tall mountains, and so you have actually more connectivity going this way than going across the Appalachians. So of course, elevation is one of the most important things. Road distance is another important thing. Human density is an important thing. Canopy, impervious surfaces, and slope. So these are the variables that are the most important in terms of tick dispersal, at least as far as the model is finding, okay? So then finally, what we do is that we have this, what I mentioned, the evolutionary genomic kind of a framework. So this is a very simplistic framework, right? So remember, the idea is to build a model that we can actually parameterize in some way. So we don't want a model that is so onerous to parameterize that we cannot actually get the data to parameterize it. This is a simple model that is just looking at what is called evolutionary fitting, right? Now it will become clear why we had to standardize all those rasters, right? Okay. So what do? We have these so-called like these loci, and we have L different loci. We use, I think, 1,000 or 5,000 or something, I forget now. But basically, each of these loci are what are called a quantitative trait locus, right? And so what is a quantitative trait locus? It's basically, for example, if you have a large number of SNPs, and each SNP has a small effect on a particular variable, then all the SNPs together will determine the phenotype of that particular animal, right? So in our case, what are we looking at? We're looking at how suitable a particular environmental variable is for that particular tick, right? So we start off where all of your environmental variables are kind of, so these are all like normal distributions, right? Because if you have a large number of SNPs, all of them are 0 and 1, right? Then you basically, across the whole population, the distribution of the trait will be normal, right? So most of the population will be at the mean, and then there'll be few at the tail, okay? So all of these, so when we start the ticks running, right, each of these traits is a normal distribution centered at 0, right? So once, but there are going to be ticks which are at all the extremes, right? Okay, fair enough. Now, when a tick is in a particular kind of a raster cell, right? So for example, so you have a distribution of these variables. So this is where it starts off at 0, right? And you can have two different raster cells. One raster cell has a value of 0, and say one raster cell has a value of, say, 10, right? And this could be any variable. Don't worry about what the variable is. It could be vapor pressure or whatever, right? But these are all standardized, right? So as I told you right in the beginning, the 0 is the ideal for that tick, right? And all these ticks start off with a value of 0. So if I take a tick with this genotype and put it in this raster cell, right? I can take how far this genotype is from this environmental value, right? And that will be approximately 0. And so this tick is ecologically fitting, right? So basically, it fits well with this particular environment. However, if I take this tick and put it into this is approximately like 10, and so it is not very fit, right? So the further you are from the 0, the less fit you are, right? So we have this kind of an exponential fitness function which determines your mortality, right? So basically, the further you are from 0, the more your mortality. The closer you are to 0, the less your mortality, right? So yeah.
So just to clarify, basically you have done a kind of a niche model for the vector, and you have an environmental gradient in space, how climate or whatever is changing. And you're trying to say that you're not tracking habitat. You're saying, suppose the vector has to get from location A.
So this is an individual-based simulation.
Okay, it's a
So
simulation.
we have tens of millions of individuals, right, which we start off with.
Okay, so they're moving.
So
they go from one cell to another cell. When they go to the next cell,
the likelihood of you going from one cell to another cell is determined. So we'll get to that in just a bit. But basically this is an individual-based simulation, right? So basically, so I have like these multiple cells. I have multiple individuals in each of the cells, and my mortality depends upon whether for each individual, whether that individual is fitting for that environment or not. And remember, this is only a single environmental variable, right? So the tick has to be fit across multiple environmental variables to be surviving, right? Okay, fair enough. Okay. So in this case, you can see that when we start off, most ticks, so if we say that this is the thing, if it's one standard deviation away, so this will be like +2, and this will be -2. So you can imagine that 10 would be somewhere here, like way past that 95% tail, right? So the likelihood that I will get by random chance a tick with a genotype of +10 is very, very small, but it is there. There's some probability that by random chance I will get a tick which has a genotype which has a value of 10, right? Okay. So this is where we start the simulation running, right? And what you do is that you have this, so you start all your loci off at 0, right? And you start the ticks only within the cells that were present, where the tick was present in 1930, right? So once you have that, you start off with by populating all the cells where the tick was present in 1930, all the genotypes starting at 0, right? And so this is the ideal kind of situation, right? So the ticks will follow the exact same distribution as that initial species distribution model predicted, right? So the areas which were perfect for the tick is going to be the best, or the ticks are most likely to survive. The areas which are not good for the ticks are not going to survive, right? So that makes a lot of sense. Then after that, we allow the ticks to disperse, and the dispersal will follow this, right? So basically, cells which are highly connected to each other, the ticks are more likely to disperse. The cells which are not connected to each other, the ticks are less likely to disperse, right? Okay. After the dispersal, the ticks then end up, all their various genotypes, whatever it is, they end up in their final cell, and then they have sexual reproduction, there's a recombination and everything else, and you set up the next generation. Yeah.
Sorry. Could you remind me what was you have like random SNPs? That's also simulated, or no? Sorry, so what is the genotype fitness?
So actually, I wanted to do the SNPs, but it was too computationally expensive, because so basically, if I have say 1,000 loci, right, and I want 1,000 SNPs per locus, then I have to basically have almost 1 million SNPs, right, and that becomes messy. But for the most part, the results will be similar, right? So for example, because it's a normal distribution, so if, for example, a male with a average value of 0 mates with a female with an average value of 10, the offspring will be a normally distributed, will be from a normal distribution with an average value of 5, right? And the same standard deviation, right? So we assume that the standard deviation doesn't change. That we do assume that, like the ticks are being produced from that same basic normal distribution. But the main thing is that if, for example, this female mates with a male, which is at, say, 5, right? Then the offspring will be from a distribution which is 2.5, and therefore, you are more likely, if you just pick randomly from this distribution, to get a value of 10 than if you pick from this distribution, right? So that's how the evolution happens, right? So basically, what happens is that when you have this mating that's taking place, you get these new recombinant ticks, right? Which may be less adapted or more adapted, but once a mortality happens, only the adapted ticks will continue to survive in that cell, right? Then again, you have the dispersal, you have the mortality, and you go on. So every generation, you have the selection, and the ticks which have a genotype which is kind of ecologically fitting to that particular cell they're in are the ones which are going to be surviving, and only the ones that survive will reproduce, and therefore the thing keeps going. Yeah. Yeah. So this is the reason why I need what do you call it? The genomic data. We ran it for 85 generations, right? Because we know that it's the tick. Sorry, we ran it for. I don't remember exactly, but we ran it for like approximately like 85 years, which I think like 40 generations, because each tick takes about 2 years for its full life cycle to finish. Yeah. 1 generation equal to 2 years, yes. Yeah. So, yeah. It takes a long time for these ticks to finish their
generation is 2 years.
Yeah, yeah,
yeah. Lifespan or generation?
Generation time.
That's crazy long.
Yeah, yeah, yeah. So, sorry? I don't know whether they hibernate or not. Yeah, but anyway. So, but again, so this is where we still have to do a lot of work, because we ran it, and I'll show you some of the results, but there's a lot of parameterization that goes into this model, and that's where I think I'm having a bit of issues. I'll come to the weaknesses of the model in just a bit. Yeah.
How far was the standard deviation from the mean? Because that also I think matters.
Sorry?
How far was the standard deviation from the mean? Because that would matter.
I want to say that we used a standard normal distribution with a standard deviation of 1 and a mean of 0. That's what we started off with. So basically, like 95% of the things will be between -1 and +1. So you cannot move like 10 in a single generation. You have to move slowly. But there is always so these are normal distributions. So it basically goes from -infinity to +infinity. So, but the probability of getting these extreme genotypes is very low. But the point is that since we have tens of thousands of ticks in each cell, right? You always have some chance that you'll get these very extreme kind of genotypes, right? And because you have sexual reproduction, the alleles which are good for one could be, so you can get these locally adapted gene complexes. So basically, you can get a situation where you have a well-adapted allele for the first environment coming together with a well-adapted like a locus for another environmental variable. So the tick becomes more and more kind of like adapted to that particular environmental condition, right? Okay. So we have dispersal, then we have mating, and remember we only allow mating within a cell. Of course, this is also a bit of a random kind of thing, because is there, I mean, these cells are pretty large, they're like 10, no, I think they're 10 square kilometer cells. So is there really going to be like panmixia within a cell? We don't know, but basically the smaller you make the cells, the more computationally intensive the model gets, right? So there are lots of things that we still have to work out. But the idea is to kind of give you the basic principles right now. So then once the mating happens, you get this kind of a new set of ticks which are now have undergone like recombination and everything else. So you have a huge diversity of these ticks, right? And remember, these are ticks, so each female can produce thousands of offspring, right? So you have 4,000, 5,000 ticks in each cell, and then you have thousands of offspring. So you have this huge kind of standing genetic variation on which natural selection can act in each generation, right? Okay. So let's just go into the results and see how the model is doing. So I just wanted to remind you, so this is the dark green are the established for 1930, the light green is established for 2015. So these are places that we know the tick is. And then the light yellows are the places where it's been reported but not like confirmed, or we don't know for sure, right? And our niche model for 2015 said that the tick is going to be here, right? About 50 kilometers north of where it was in 1930. And how does the emerge model do? The emerge model does really well, right? So compared to our other niche model, it's saying that, well, it's over predicting. So basically it's saying that, you know, like it's going all the way. So all the reported areas are also areas that the tick is being, the model is saying that the tick is present in. But you can see that there are some areas which are definitely zeros, which the model says that the tick is not going to be present in, right? So, but the main thing is that you can see that New York, right, where the tick has really spread, is being well kind of predicted by this model, while in the original niche model, New York was zero, right? There was no presence of the tick, right? I think that for me, yeah. Sorry?
The variation in the mean in the different cells, like in the New York cells?
No, we didn't. So this is just a single model kind of realization. It takes a lot of time to run these models. So again, there's a lot of work that needs to be done, and we are still waiting for genomic data and all, and I'll tell you why the genomic data is important, but no. So this is just a probability that comes up from a single thing. And it's actually not even a probability. It is actually a standardized population size, right? So habitat suitability for this model is how big the population is relative to the population in another cell. So if it's zero, then it's blue, but if it is, as you get darker and darker, the population size, the number of ticks in that cell that are surviving that particular environmental condition is higher.
But here your 0 will become 10 and it will gradually change.
So no. So basically the genotypes are going to be moving and are going to be becoming closer and closer to that particular environmental condition. So there's going to be more and more ecological fitting, right? But different tick populations may be like evolving in completely different directions. Make sense? Yeah. So for me, again, I started off this modeling as a more of a thought experiment than anything else to just see, you know, like, is there any way that we can like improve these species distribution models, start incorporating more ecologically sensible variables like evolution, dispersal, and everything else. But kind of a side result, which I think is more exciting than anything else, at least as far as I am concerned, is that when you're looking at range expansion, right, people have said that range expansion can happen due to two very different processes. You can have something which is called diffusion. So what do we mean by diffusion? So if I have a cell here, right, and the temperature is moving this way or whatever it is, the tick is here, right? You would assume that the tick would then go here, and then it will move here, right? So this is just a diffusion process, right? The other is that you could have what is called a jump dispersal, right? Where you again have these three cells, but the tick from here goes here, right? And it doesn't colonize that in-between kind of spot. So at least as far as Ixodes scapillaris goes, they have data to show that this kind of jump dispersal is how these ticks seem to be doing this range expansion. And we have found evidence from the model to show that this is how the model is also predicting this range expansion will take place. So this is our 1930 distribution. This is some intermediate kind of a point. You can see that there are these small populations here, and then there's this relatively large population there, right? And then a little later, you can see that this population now starts coalescing backwards, right? While this is still moving kind of, the southern population is moving north, the northern population is moving south, and then finally, the two populations kind of coalesce, right? And we actually have data from the model that gives us a mechanistic explanation as to why this is exactly happening, right? So I know that this graph probably looks very familiar because this is the same kind of graph I was using for our virulence, right? So, but this kind of a pattern is very common in biology, right? So what the model data is showing, again, we don't know whether this is a reality in the natural world or not, but this is what the model is predicting and showing. So basically, if this is the source population and that is some very distant population, right, you can see that the number of immigrant genomes, right, if you just look at distance, would be increasing. So it would be increasing as these populations become closer and closer, right? Now the problem is that as the number of immigrant genomes increases, your likelihood of invasion does increase, right? There's no problem there. The problem is that when you have a lot of these immigrant genomes coming in, any population there that is getting locally adapted gets swamped out. So you get this breaking up of these locally adapted gene complexes, right? So what happens is that having too many immigrants is good when you are trying to invade, but it's bad when you're trying to adapt, right? So what happens is that your establishment starts reducing as the number of immigrant genomes increases. So what happens here is that in number one, you have a lot of immigrant genomes coming just simply because it's so close, right? Regardless of the connectivity or regardless of anything, a cell which is right next to another cell is going to have a lot of ticks coming in, right? But the ticks that then adapt to that particular condition. So after the mortality happens, right, there's going to be a set of ticks that are now adapted to that particular new condition, right? But then there's dispersal that happens before the mating happens, right? So the ticks that have now adapted are a very small subset of ticks, right? Because there's a huge mortality, right? So there's a small subset of ticks which is adapted, but then there's this huge number of new like immigrant genomes coming in which are not adapted, right? So they just completely swamp out all of this adaptation that's happening, right? When you go to number 3, the problem there is that the likelihood of you even getting enough new immigrant genomes to have adaptation is so low that you never get establishment, right? Number 2 is the kind of sweet spot, right? You have enough immigrant genomes to come in that you may by random chance get something that is adapted to that location, but you don't have so many coming in that once you have an adapted genotype, that it's just going to be swamped out by completely unadapted ones, right? So you start seeing this kind of establishment that's happening here. And then, of course, once this population, this population then grows by diffusion, right? So that's why you see that you have this thing, and so this population is growing by diffusion here, so it's moving up. This population growing by diffusion here, but the big jumps are coming through this kind of jump dispersal.
Dispersal is happening through humans.
We don't know. I mean, the model is not very explicit about how
Yeah. But I'm just curious if there is a possibility of...
Humans are probably a big factor, yeah. So for example, when we look at this, so distance to roads is one of the most important variables, right? So humans are definitely a major factor that is moving these ticks around in the landscape. So elevation is one. So I had expected that it'll be forests or something of that sort. It's like roads. So and deer are definitely not walking along roads. So
positively correlated.
Roads were positively correlated.
So low resistance, basically.
Sorry?
Low resistance.
Low resistance, exactly. Okay. Okay, yeah.
When you're mentioning the term invasion, are you speaking like how do you define that invasion with respect to this particular slide? Like, are you trying to say that your tick is invading or?
But that's a very good question. I'm using these terms kind of loosely. So invasion just means that the tick arrives in that particular raster cell.
Okay,
right?
Wouldn't...
and this is before the mortality. So this is before the selection.
Yeah, but wouldn't be it more appropriate that we could use this word colonization rather than invasion?
Colonization would probably be a good term.
Because invasion is something when you consider with respect to a native or resident species.
Fair.
You look, yeah. So that.
Yeah, we can definitely use the word colonization. That may be a better term. But basically, as the number of immigrant genomes increases, you just have more likelihood that a tick is going to arrive in that particular place. When you just have few genomes, then you can't even arrive, right? So that's the basic idea. Okay. Now, I think that this for me is probably the most exciting part, and that is that as I told you before, since this the model has this kind of evolutionary genomic framework, of course, it's just quantitative loci. But basically, what happens is that even with these quantitative loci, we can actually calculate all the genetic statistics, right? So we can calculate FST, we can calculate everything, right? So what I did was that, so for example, this is a map of this GST. And this basically, it's a very simple thing. So the lower your value, the more genetically related the two cells are. And the higher the value, the more distant the two cells are. And remember that all of this is a pairwise comparison. So I'm comparing it all to this black dot here, right? So basically, you can see that all of the cells here have very little genetic differentiation. And then as you go more and more north, there's a huge amount of genetic differentiation. And so this is a variable. So this is basically, I think this was the vapor pressure, the locus that was associated with vapor pressure. And you can see that there's a very strong selection. So basically, as the tick moves north, there's a very strong adaptive kind of signature of adaptation that we are seeing, right? While at the core of its range, there's no differentiation at all. And this makes a lot of sense, right? So there's a selection gradient that we can pick up, okay? But this is the really interesting part. What you find is that, so we also have a bunch of neutral loci, right? Which are not associated with any fitness benefits whatsoever, right? And you can see that, fine, the strength is definitely less, but you do see that there is this huge genetic differentiation that you see between this one and this one right there. Why do you think it's a neutral locus, right? So why do you think you're actually having genetic differentiation happening despite the fact that it's neutral? It's not really being selected. Farah also talked about the reasons for this.
So when do you have genetic differentiation? Remember Farah was talking about that island or isolation by distance, right?
Yeah.
So if you have isolation by distance, you have one population and you take some of them or whatever, like there's some distance. Would you have genetic differentiation between those populations?
Just by random chance, genetic drift.
Because, for example, if there are 4 populations, if you compare population 1 to 4 versus 1 to 2 or 2 to 3 or 3 to 4, right? So 1 to 2, 2 to 3, 3 to 4 should be, say, different by x. Then 1 to 3 would be maybe 2x, right? So you can have genetic differentiation by distance and gene flow, something like that, yeah?
Yeah, exactly. So this is exactly what Uma was saying. So what we are finding here, right, is that there is this strong genetic differentiation due to just random genetic drift. But the most important part is that the genetic differentiation is actually strongest at these expansion fronts, right? And this is what is called allele surfing, right? And this is well known in many of these species which are rapidly expanding. So what happens is that because these new populations have such small founding population size, you have huge amount of genetic drift that's happening there. Once they start kind of stabilizing, then this neutral genetic variation starts reducing, right? So you start seeing this. But from my perspective, right, it is really important because, so for example, right, if I was to do, say, a genome-wide association study, right, and I'm trying to identify the genes that are really significantly associated with, say, adaptation to vapor pressure or something of that sort, right, I will be using at a SNP level the same variable GST, right? So basically, the SNPs that are highly significantly differentiated are the ones which are likely under selection. The SNPs that are not differentiated are not being selected, right? So that's what I'm going to be using. But the problem is that you have a confounding, right? So you cannot use your standard genetic, your standard GST, right? You have to correct for the fact that the areas that are undergoing the strongest kind of selection are also going through a whole lot of other factors. So you could have these kind of completely artificial, so basically you could have a SNP which is completely neutral, right, which has a huge GST, not because it's under selection, but because of these founder effects and other things, right? So all of this GST and everything else assumes a neutral population theory, right? So equilibrium theory, right? But these are not in equilibrium at all. These populations are not in equilibrium, and so you have to reimagine how you're going to be identifying SNPs in these rapidly expanding populations. Sorry, go ahead.
No, just to say that, so imagine you had 4 populations, right? The 4 states or 5 states, and imagine that they were in equilibrium. So, you know, that population 5 would be up there somewhere, and obviously it would be very different from population 1, which was down there in, was that Florida?
Yeah, Florida, that's Florida.
Right? But imagine now instead, you only had 3 populations, but 4 and 5 have been colonized from 3.
Correct.
So some individuals from 4 went to, some individuals from 3 went to 4, and some of those individuals from 4 went to 5. So you're having these bottlenecks. So when you have these bottlenecks, there'll be only small amount of variation. But whatever that variation is, might be quite distinct from the variation in 3. So it look like they are very different,
Correct,
as if it was 1, 2, 3, 4, 5 right from the beginning, or they're more different maybe because you'll infer that, oh, because 5 is much hotter or something like that, colder or something like that. But in reality, it's just because of, you know, the process of colonization. So basically, that's really important, that you are inferring all kinds of things like, oh, maybe it's much colder there and the ticks have completely adapted and all that. But it's actually just because of what's called allele surfing, which is basically colonization processes, expansion, population expansion.
Perfect.
And that's going to be very common in vectors.
It's going to be very common in vectors,
Yeah, it's very interesting how in the morning, Kavita talked about, you know, you need to have mechanism. You can't just make predictions, okay, it's warming, so vectors are going to expand. And Farah was showing how you can actually use genetic and genomic data to track those expansions, track those changes in population size, how populations are related to each other. And now Guha is talking about these kind of an integration of both those. I hope you're seeing that.
Wonderful. So finally, I just wanted to mention, yes, Ashwin.
In Georgia, just 7 o'clock from your that circle point, why is there a red spot there? Why is that GST is higher in that spot? Is it anything geographically distinct there?
I have not looked. I'll have to go and look.
It's elevationally
I think that there is
same.
something to do with the fact that the tick is actually not present there also in the beginning. See, this area it's not established. So I don't know what's happening there. But there must be something, the environment must be quite distinct. The appellations do come all the way down. So it may be that it's part of the appellations or something of that sort. But it's a good point. I didn't really look. Yeah, but it's a good point. It's right there. Yeah. Okay. Finally, I just wanted to mention that, see, so because the model makes these very explicit spatial predictions, I can actually say that, okay, fine, what is the genetic distance between, say, New York, I don't know what ME is. Maine? Yeah, Maine and South Carolina and North Carolina, right? So the interesting thing is that a paper came out by Monzon et al. In 2016. And so this is the prediction from our model. And Monzon's paper was basically was finding a very interesting pattern that when you looked at Amblyomma americanum, right, that the main population, which is all the way over here, is actually intermediate between the New York population and the South Carolina population, right? And this was kind of a very surprising finding because it was always thought that, when you looked at this genetic differentiation, it would be North Carolina, South Carolina, New York, and then the New York population would be colonizing your Maine, and so you would basically have this kind of isolation by distance rather than having Maine right in the middle. But this is exactly what our model is predicting too. And the reason for that is actually very simple. It is that New York has very high connectivity, but the bifurcation actually happens right here. So if you look at our resistance map, right, you can see that when you look at the resistance, you see that there's one path that goes here and then another path that goes here. So there's a bifurcation at New York. So if most of the ticks are colonizing from South Carolina and North Carolina, which is the core of the distribution, right? You would find that that Maine would be somewhere in between because it gets some ticks from New York, but it also gets a lot of ticks from North Carolina and South Carolina. So you can see that, at least for me, what was exciting about the model is that unlike most species distribution models, it makes predictions, it makes specific spatially explicit genetic predictions, which can then be validated with data that is not used to fit the model, right? So I can go out, I can collect genomic data from these ticks across their range, and I can actually ask, how well is this model predicting? The minute that I can do that, I'll have a little more confidence that this model is actually doing a good job, so that when I'm predicting for the future, I'll have a little more confidence, right? As I mentioned in my first class, all models are wrong, some models are useful. So the idea is that, will we ever know whether these models are correct? Probably not, but the more kind of lines of evidence that we have to say that the model is accurate, the more confident we are that our predictions are indeed correct. Yeah.
How was the data collected, and is there any difference in the data generating process in 1930 versus 2015?
Oh, that's a great question. I don't know. So this was a paper that came out in 2015, and it was basically telling us, okay, fine, these counties have Amblyomma americanum, these counties don't have Amblyomma americanum. And again, it wasn't collected data. It was just kind of compiled from a large number of papers that were reporting it in these different counties. So is it the most accurate data? Probably not, but it's the best data that we have. Okay. So, of course, lots of caveats. So I think that the first one is that we have to do this sensitivity analysis. So we have to know, how changing these different parameters affects the model itself, right? So right now we've tune the model so that it fits this data well, but we don't know how these different parameters are affecting. We do need better temporal resolution so in terms of the tick range expansion. For right now, we only have like 1930, 2015. So we don't have any in-between points. And so there's no way of us having any in-between points. Ideally, we would have at least 3 points, right? So if we have a 1930, and then we have some in-between point, we can fit the model, we can, with the 1930 and the in-between point, and then we can predict to the current, then we would have real evidence that the model is working. But right now, we only have the 1930 and 2015, so that is another major issue. The final and the reason that this model remains unpublished today is that I'm still waiting for this kind of tick genomic data at the kind of spatial scale that we need to validate this model. There are a bunch of people who are working on Amblyomma americanum. The Amblyomma americanum gene has been sequenced by a bunch of people that I've been working with. I'm not directly associated with the genome, but the minute that the genome is there, the hope is that they'll get the funding for doing a population genomic analysis across the whole scale. It's a very medically important tick right now, so it is likely that we will get this. I think that once we can get this and we can actually demonstrate that the model predictions are indeed correct, then I think I'll be more confident about publishing this model. As of now, it's just a thought process, but I think what I wanted for this particular talk was to say that, all of us need to be like kind of moving out of our comfort zones, right? So, in the first talk, that was perfectly within my comfort zone. It's something that I've been doing for a long time, and so I was kind of thinking, this is, you should be willing to put yourself out there, saying that, hey, I actually don't know what's going on. I don't know whether this is correct or not, but it's an interesting idea, and like, what do you all think about it? Maybe it'll go somewhere, maybe it won't, and that's how science progresses, right? So, so that's about it, and I will take questions. Yeah.
Yeah. How important is this particular disease for their government from a public health perspective? How important is this?
So, Amblyomma americanum is a very important vector. It transmits a whole lot of vector-borne diseases, but as of now in the US, it's becoming really kind of important because of this this red meat allergy that it's causing. So red meat allergy is not an infectious disease, but it is caused by this tick. So there is a
for the government. It's a priority.
So surprisingly, I know that this is probably a bit controversial, but NIH surprisingly has not made tick-borne diseases a great priority, despite the fact that most deaths due to vector-borne diseases in the US is actually due to tick-borne diseases, not mosquito-borne diseases. But a large proportion of NIH funding still goes for mosquito-borne diseases, but that's because it causes a huge amount of mortality in Africa. So tick-borne diseases have always kind of taken a backseat to mosquito-borne diseases. I think mainly because they're more regional specific, the amount of mortality may be less. I'm not really sure, but generally tick-borne disease are more disease. The other aspect, which I have to admit, is ticks are much more difficult to work with as compared to mosquitoes. Mosquitoes have a generation time of what, 21 days, right? So you can get them into the lab, you can do experiments on them. Everything is much easier as compared to ticks. For example, mosquitoes, I can actually get them into the lab. So during my postdoc at NIH, what we did was we went out, we collected these mosquitoes from these 8 different natural populations. I was working with Aedes albopictus. We put them into containers and we just FedEx them across, so with all the bio containment and everything else. But then basically I just FedEx them to NIH. Once we got to NIH, the first generation, we had to kind of feed on these lab animals. But after that, right, we could move it completely to, like this membrane feeders and everything else. So mosquitoes can be easily domesticated to kind of, to be a completely in vitro system without any animal systems. Ticks are practically impossible. right? So they've been trying to develop, I think a few labs have managed to optimize a system of membrane feeding for ticks, but ticks, they attach, right? So they have this very long attachment process. They feed for weeks at a time. So there's a huge amount of process that goes on in the case of ticks, which is not the case in mosquitoes. So ticks are just a lot more difficult to work with as compared to mosquitoes. They also have 3 life stages. Each of the life stages is completely different from each other. You know, so it's a much more difficult system. Yeah.
So if I'm not wrong, the east coast of US is more prone to hurricanes. So that would cause a kind of pulse pressure on the ticks. Is that incorporated in the model, or how would that change?
No, not at all. So actually, I only fit the model for the East Coast. The model fails quite miserably when it starts going to the West Coast. I'm not really sure why. So that's another thing that we have to look at. So yeah, but we also don't have very good data on how amblyoma spread in the West Coast, whether it was like where it was originally present and where it went. So that's another issue. But we focus on the East Coast because that's where the data is really good. Yes, Ashwin.
Genotype data is from what exactly? I mean, it's a single gene or something? Like just
Which genotype data?
You use the genotypes, right, in one of your models?
No, this is actually the data that is generated by the model itself.
Okay.
Yeah. So basically we have this evolutionary genomic framework with these multiple quantitative trait loci, right? And so this is the GST. It's based on a simulation, completely simulated.
And
There's
for
no
the landscape resistance?
I think we simulated like 1,000 loci, and of which I think 15 or so were under selection because we only had 15 environmental variables.
And for the landscape resistance, the ticks are not completely free living animals. So
yes.
Them choosing, to certain route, they are not really flying also like mosquitoes.
Very true.
So how did you get into model?
We didn't. So that's where it's still a correlative model, right? So Farah was talking about the fact that we have to move to more mechanistic models and everything else. The problem with mechanistic models is that they are very, very difficult to parameterize. The amount of data you need to parameterize a mechanistic model is very, very large. These correlation models that way, they just fit data, right? So whether or not they're real is a little unclear right now, but at least you can fit the patterns that you're seeing, and then they're useful for prediction. Great. Thank you so much. But yeah, so if anybody has any questions about the model, I'll be happy to talk to you about it.
Yeah, so actually what Guha showed is actually a very interesting and different approach that we have not heard of so far, which is individual-based models, right? So each of those ticks is a unit in that model, and each of those ticks has associated with it, like a it's like a data structure, has many different characteristics, like it has a genotype.
Yes.
The genotype has 1,000 SNPs. So it has, imagine 1,000 columns, okay? And basically of those 15 are under selection. So basically there's a equation which governs what change happens in them related to the environment or the cell in which that animal is. So those kinds of things. Basically, like, Kavita was talking about, okay, how exactly will temperature impact vectors, for example? Okay, will it make them bigger, smaller, whatever, reproduce more and stuff? And this is another way of thinking, okay, suppose we model this. We try this, we try that, we try something else. What actually fits the data? So it is also correlational in some sense, but it is involving a mechanism in that correlation. Not just saying, overall we find some change, and so there's, I don't know whether I'm...
No, you're saying it exactly right. So basically, if you have this, and this is one raster cell, right? That raster cell will basically have a 3-dimensional matrix, right? And this 3-dimensional matrix will be basically your ticks here, So basically, this is tick 1, 2, 3, 4, 5, right? And then this is a single locus, right? Locus 1, 2, 3, 4, and then these are the kind of the SNPs for that. So basically, each of these is going to be, so this will be each tick, right, will have multiple loci, and each locus will have multiple SNPs. So that's where we are kind of like going with this. And then so this is just one population. Then the next cell will have a different population. And then remember that this particular cell will have a certain environment. This particular cell will have a certain environment. And you're asking how well or which of the ticks in this matrix are going to be fitting this particular cell or this particular environment, which of the ticks in this are fitting this environment, right? And then you have dispersal between the cells, right? And then after that, you have mating between the ticks, right? So it's a pretty as Uma said, it's an individual-based model. So it's not the most efficient way of doing it, but this is the only way that we could think about how to bring in evolution into this kind of system while still having it being tractable enough that we can build it with the data that we have at hand.
Write the simulation, or have you used a simulator?
No, we wrote, it's an R.
Oh, you wrote it's an R. Because there's something called, landscape genetics people use called POP something, and I'll remember, but actually you can have loci which are potentially under selection. And the other thing is there are many population genetic approaches to analyze SPLAT-G and stuff, to analyze gene environment correlations in the context of landscape. So,
no, I didn't know about this. So yeah, maybe this is a bit of overkill, but there are lots of things that I do which are just to learn something new. So yeah, but I'll definitely check it out if you can find out the name. Thank you. Thank you so much. Thank you.